I wanted to thank the organizers for inviting me to give this talk about senescence and age-related osteoporosis and frailty. I'll cover some of the work in this area that my group and others have been doing over the past several years. So I have no relevant financial disclosures. So I want to start by just talking about the remarkable progress that's been made over the years in terms of the treatment of osteoporosis. So you can see 25 years ago, essentially when we would go to the osteoporosis clinic, we would offer estrogen to women and calcium and vitamin D to men. Now, if we fast forward to 2022, we don't really use estrogen, and raloxifene is not used much, but we have 4 bisphosphonates, teriparatide, abaloparatide, denosumab, and romosozumab. Odidacatab, unfortunately, did not make it into clinical practice. And if you look at these drugs, the paradigm for drug development has shifted from observational or opportunistic to pathway-based discovery driven by advances in fundamental bone biology. And in particular, for example, in the case of romosozumab, clues derived from rare bone disease patients. So this is really, I think, a great example of how investing in discovery science has in fact translated for patients into novel therapeutics. So with that, with these options that we have, what is the case for new approaches to treat osteoporosis? The first is that it remains a huge public health problem. It's not well appreciated that the number of women who will experience a fracture in 1 year, in fact, exceeds the combined number of women who will experience incident breast cancer, myocardial infarction, or stroke across all ethnic groups. So osteoporotic fracture is, in fact, a huge clinical problem. And there are important gaps in the current therapeutic arsenal. So because of the fear of rare side effects, in particular, osteonecrosis of the jaw and atypical femur fractures, use of bisphosphonates in particular has become limited. So I think we need newer agents that lack these complications. The efficacy of bisphosphonates beyond 5 years still remains unclear, so we don't have the perfect long-term anti-resorptive drug. There's also a great need for new anabolic drugs. So if you look at teriparatide, abaloparatide, or romosozumab, the stimulation of bone formation, or the anabolic window, if you will, is limited to 6 to 9 months. And for reasons unclear, the ability of these drugs to indefinitely stimulate bone formation wanes after that period of time. But beyond these issues that are specific to osteoporosis, there's in fact a broader fundamental problem with current treatments for osteoporosis and really all other chronic diseases. And this is highlighted here. So the current treatment strategies for all chronic age-related morbidities are disease-specific, with each drug targeting a single disease. So if you're a 70-year-old man or woman, for example, you might take a statin to lower your cholesterol, a antihypertensive for your blood pressure. If you happen to have diabetes, an antihyperglycemic for diabetes, a bisphosphonate for osteoporosis, and so on. So the problem with this is polypharmacy. It's estimated that 67% of older adults take 5 or more medications. And this inevitably leads to an increased risk of side effects, adverse drug interactions, and non-compliance. Now, specific perhaps to osteoporosis is that the busy primary care physician is often dealing with perceived more pressing immediate medical problems, such as the blood pressure of 170 or the blood sugar of 250. So the T-score of minus 2.6 drifts down the problem list where it may not get addressed for a long time. So it's in this context that we as endocrinologists, and me as an endocrinologist and a researcher in osteoporosis, thought about osteoporosis in isolation. But I think it's important to think about osteoporosis in the broader context of all of the aging-associated comorbidities that accompany osteoporosis. So many patients with osteoporosis have diabetes, dementia, renal failure, vascular disease, and so forth. And the common risk factor to these, in fact, is aging. These are all age-related comorbidities. And this recognition has led to this geroscience hypothesis, which says that manipulation of fundamental aging mechanisms will delay in parallel the appearance or severity of multiple chronic diseases, because these diseases, in fact, share the same underlying risk factor, which is aging. So in terms of aging, there was a landmark paper back in 2013 by Lopez Otin and colleagues who identified 9 fundamental hallmarks of aging. These hallmarks were kind of built in a pattern similar to a previous work identifying hallmarks of cancer. And you can see the list here. Many of you are familiar with this. For example, telomere attrition, epigenetic changes, mitochondrial dysfunction. In my talk, I want to focus in particular on cellular senescence, which has received considerable attention by many groups around the world. So what is cellular senescence? It's not a new concept. It was actually described by Dr. Hayflick in 1961. It's a cell fate that involves essentially irreversible replicative arrest, activation of tumor suppressor genes, chromatin changes, resistance to apoptosis, and often increased protein synthesis. Now, the proteins that are made are these so-called, as I'll come back to, senescence-associated secretory phenotype, or SASP, which is really a sterile inflammation that causes tissue dysfunction. So this is Dr. Hayflick, and he described back in 1961 that when you cultured, for example, mammalian fibroblasts in culture, they grew, but then eventually they stopped growing and entered what he called senescence. Now here they didn't die. In fact, they were resistant to apoptosis, but they just sat there doing nothing. And that was the original description of senescence. Now it's now clear that senescence occurs in vivo, and there's a lot of documentation of that at this point. So the idea here is that with aging, due to cellular damage, you get an accumulation of senescent cells. Now, as I'll show you, you don't need a lot of senescent cells. Maybe even in very old individuals, 10 to 12% of the cells become senescent. But they cause tissue damage by the secretion of this senescence-associated secretory phenotype, or SASP, which consists of cytokines, chemokines, metalloproteinases. So these accumulate with aging at sites of age-related pathology. And the hypothesis is that if you target these cells and either kill them or prevent the secretion of the SASP, that you may be able to have a rejuvenating effect on aged tissues. Now, in recent years, the molecular mechanisms driving cell senescence have been well worked out. It's known that when a cell is exposed to various insults, such as DNA damage, oncogenic mutations, reactive metabolites, mitogens, and so forth, that these types of stresses to the cell lead to activation of 2 key pathways. One is the P16-INK4A pathway, and the other is the P53-P21 pathway that ultimately result in senescence. Now, one of the characteristics of senescent cells is upregulation of these SCAPs, which is the senescent cell anti-apoptotic pathways. So these are a family of genes that confer resistance to apoptosis to these cells. And in fact, it's these genes that are targeted, as I'll show you, by the so-called senolytic drugs, because if you can target these genes, you might be able to overcome their resistance to apoptosis. Now, these triggers also lead to a DNA damage response with activation in particular of NF-kappa B, which leads to the secretion of a number of pro-inflammatory cytokines, and ultimately the SASP, which then leads to tissue dysfunction. Now, as I mentioned, senolytics are drugs that target these pathways that are upregulated in senescent cells and confer resistance to apoptosis. So these drugs relatively selectively kill senescent cells without affecting normal cells. The other way to target senescent cells is not to kill them, but to prevent the secretion of the SASP, and these are the so-called xenomorphics. And one example of xenomorphic drugs are the JAK inhibitors that are used clinically for inflammatory diseases like rheumatoid arthritis and hematologic malignancies. These drugs don't kill the cell, but they inhibit the production of the SASP. So I'm going to focus on P16-INK4A because that is a major trigger for the induction of senescence. So a number of years ago, we asked a very simple question, starting in mice, which is that, is there an increase in senescent cells in the bone microenvironment? So here we looked at osteocyte-enriched bone samples with aging from mice, and you can see this is in female mice, and this is the P16-INK4A messenger RNA as a marker for senescence. And it remains relatively low until about age 18 months, and then it goes up dramatically. This is in male mice where you start to see an increase a little bit earlier, around 12 months, 18 months, but then again, it's highly upregulated by 24 months. We also looked at other cells in the bone microenvironment. So I just showed you some data on osteocytes, but we looked at osteoprogenitor cells, osteoblasts, B cells, T cells, and myeloid cells. And we isolated each of these cells from the bones of the mice without culturing the cells. So we examined them for expression of P16 right after isolation. So you can see in all of the cell types in the bone microenvironment, there was evidence for markedly increased expression of P16-INK4A as a marker for cellular senescence. P21 seemed to go up modestly in the osteocytes, but didn't change much in the other populations. Now, in order to prove senescence, you also have to show evidence of what's called irreversible telomeric DNA damage. And one of the assays for this is the senescence-associated distension of satellites. So here's an osteocyte nucleus from a young mouse and an old mouse. And you can see in the osteocyte from the old mouse, this distension of pericentromeric DNA. And this can be quantified as a marker for senescence. And you can see if you look at the percentage of senescent osteocytes, it goes from about 2 or 3% in young mice to about 12%. And this 12% is actually quite consistent across tissues. So if you look at the skin of old monkeys, for example, they have a senescent cell burden of about 10 to 15% in this range. But these cells, as I mentioned, secrete the SASP, and that's how they cause their tissue dysfunction. Now, more recently, we've done another measurement. It's called telomere-associated foci. So here we use, again, this is an osteocyte from a young mouse and an old mouse. Gamma H2X is a probe for DNA damage, and that's green. The telomere probe, which is the end of the chromosome, is in red, and where they overlap in yellow is where you have a site of telomeric DNA damage, or TAF, which is a hallmark of senescence. And you can see the mean number of TAFs per osteocytes goes up significantly with age. And then if you look at the percentage of TAF-positive osteocytes having either more than 1, more than 2, or more than 3 TAFs, they all go up with aging in bone. We also looked at the SASP by qPCR, and you can see this is a heat map. So red indicates higher expression of particular genes. So for example, CCL5, it's all blue in the young mice, and then it really goes up in expression in the old mice. So this is quantified here, but you can see there's a marked upregulation of the SASP, or the pro-inflammatory secretome, associated with senescence from osteocytes in old mice. The other population where there's a marked increase in the SASP, it turns out, is myeloid cells. So these are the CD14 positive cells in the bone microenvironment. It's low in the young mice, and you see this marked upregulation of the SASP quantified here. Now, that was in mice. We also looked at humans. These are tiny needle bone biopsies that we get, and this just shows a micro CT showing the trabecular and the cortical bone. And you can see in the human bone biopsies, both P16 and P21 went up significantly in the bones from the old as compared to the young women. And then when we look at SAS markers, you can see a significant number of these SAS markers go up in the biopsies from the old women. So these studies then demonstrate that with aging, there's in fact an accumulation of senescent osteocytes, as well as other senescent cells, in particular senescent myeloid cells in the bone microenvironment. these cells have high expression of multiple SAS markers, which, based on what we know about these pro-inflammatory cytokines, could lead to an inhibition of bone formation and an increase in bone resorption. Oops. So that was kind of describing the changes that occur. How do you prove causality? So to prove causality, you have to intervene. And for that, we took advantage of this mouse model. So this is the INCATAC model, where the P16 promoter, and remember, I mentioned P16 goes up in senescent cells, drives the suicide gene in senescent cells. So this is the INCATAC model, where you have a FKBP caspase 8 fusion protein that goes to the cell membrane, but this is inactive. When we give these transgenic mice that have this transgene, so they upregulate this gene in senescent cells, when we give them this drug, AP2, this is a synthetic drug, it causes dimerization of the FKBP binding protein and activation of the caspase, killing the cell. So basically what this does, it allows for inducible elimination of P16 positive senescent cells when you give the synthetic drug. Because as I mentioned, it's the senescent cells that have high P16 expression that are going to express this transgene, but nothing is going to happen to those cells until we give this AP drug. And this is just shown here. This is a epididymal fat pad from an old mouse. And this SA beta gal, which is the blue, is a stain for senescent cells. So this is a mouse without the AP drug, but when you give the AP drug, you can see all the blue disappears because you've cleared the senescent cells in the fat. So we used these mice and treated them for 4 months with either vehicle or the AP drug, and you can see in fat tissue, we had a significant reduction in the P16 mRNA. This transgene also carries the marker, and that also went down. When we looked in bone, there was a significant reduction in the P16 message in bone. And when we used the SADS assay for senescent osteocytes, you can see there was about a 50% reduction or so in senescent osteocytes in the bones of these animals. So the AP drug in this genetic model reduced by about 50% the number of senescent cells that were in the mice. And this had a pretty significant effect on the bone. So this is the spine micro-CT from a vehicle or a AP-treated mouse, and you can see with clearance of senescent cells, there's a higher spine bone volume fraction and some of the other spine parameters. This is accompanied in the spine by a reduction in osteoclast numbers, which would be expected to reduce osteoblast numbers because of this coupling between bone resorption and bone formation. But interestingly, bone formation or osteoblast numbers were actually maintained in these mice, indicating a favorable effect, as I'll come back to, on osteoclast-osteoblast coupling. When we look at the femur, there was an increase in the cortical thickness at the femur and a stronger femur in terms of the failure load. And as at the spine, there was a reduction in osteoclast numbers. But here, we actually, on the endocortical surface, saw an increase in osteoblast numbers with an increase in the mineral apposition rate and an increase in the bone formation rate. So not only when you cleared senescent cells, you reduced bone resorption, but you either maintained or increased bone formation, which is a very favorable profile in terms of the skeleton. Now, a related question is what happened to the marrow fat in these individuals? These are human data from 1970s from Pierre Monnier, showing that in humans that as we get older, there's a decrease, as you see in the trabecular bone volume. These are bone biopsies, and an increase in the marrow fat. So bone goes down and fat goes up in the marrow. This is in mice. So this is an 8-month-old mouse versus a 26-month-old mouse. And you can see the trabeculae in turquoise, the marrow, and you can clearly see the marked increase in marrow fat in the old mouse. Now, the significance of the marrow fat and how this adversely affects bone is still being investigated, but this marked increase in marrow fat is a very reproducible finding with aging. So we then asked the question that with aging, there's a decrease in bone and an increase in fat. Is there a fundamental aging mechanism that's driving both of these? So we went back and looked at the marrow of the AP-treated mice, and you can see in the mice that got the AP drug, there was actually in the marrow a reduction in adipocyte number, the size of the adipocytes, and the volume of the adipocytes. So we have a number of in vitro studies that have shown that clearing senescent cells alters the lineage commitment of the precursor to the bone versus the fat. But what this says in vivo is that, in fact, cellular senescence may be contributing both to the reduction in bone volume and to the increase in marrow fat that's seen with aging. Now, of course, you can't use a genetic approach in humans, and for that, you have to identify drugs. So in this work from my colleague Jim Kirkland's lab, we identified expression of pro-survival networks in senescent cells. So what was causing these senescent cells to resist cell death or apoptosis? And then these pathways were silenced and causing these cells to die. So this is a bioinformatic analysis showing key nodes in these survival pathways. So BCL2, for example, is a known anti-apoptotic gene. So then a bioinformatic approach was used to match known compounds inhibiting these pro-survival pathways. And eventually the combination of dasatinib, which is a clinically approved tyrosine kinase inhibitor, and quercetin, which is a flavonoid. So the combination of D plus Q was found to selectively kill senescent cells. And we validated this in an accelerated aging model in mice where it extended health span, prevented comorbidities, and has also extended the lifespan in these mice. So D plus Q is probably the first generation of the so-called senolytic drugs that are being developed. So we went back to the mice and treated the mice with D plus Q. Here, the DQ is given very intermittently, only once a month for 4 months. And you can see with this, there's a reduction in the P16 message in bone, a reduction in senescent osteocytes, and also a reduction in the P16 message in fat. This is the SA beta gal staining in fat. You can see the blue that goes away with the administration of the D plus Q. So when we gave the D plus Q, very similar to the genetic model, there's a significantly higher spine bone volume fraction in the mice given the D plus Q, along with some of the other parameters. Similar to the genetic approach, with clearance of senescent cells with D plus Q, there's a reduction in osteoclast number, but no change in osteoblast number. And then very similar findings at the femur, with an increase in cortical thickness, in femur strength, a reduction in osteoclast number, but again at the femur, an increase in osteoblast number and an increase in the bone formation rate. Finally, we used the xenomorphic approach and treated the old mice with a JAK inhibitor, which you might remember I told you would not kill the senescent cells, but it would reduce the secretion of the SAS. And again, the findings were very similar to killing the senescent cells, an increase in the spine bone volume fraction, and very similar beneficial effects at the femur. So what these studies showed then was that in old mice with established bone loss, 2 to 4 months of treatment with either a senolytic, D plus Q, or xenomorphic, a JAK inhibitor, improved bone mass, microarchitecture, and strength. The beneficial effect of targeting senescent cells was due to suppression of bone resorption, with either a maintenance of bone formation in trabecular bone or an actual increase in bone formation in cortical bone. In data that I don't have time to go into, in vitro, we showed that the senescent cell conditioned medium impaired osteoblast mineralization and also enhanced osteoclast progenitor survival. And one of the important interventions was when we did these interventions in young mice who shouldn't have a high senescent cell burden, there was no change in the skeleton. And that would make sense because you need to have a high senescent cell burden for any of these approaches to have an effect. So here I compare anti-resorptive versus senolytic therapy. So the idea is that the senescent cells in the bone microenvironment is stimulating osteoclastic bone resorption, inhibiting osteoblastic bone formation. When you treat with a bisphosphonate, you basically kill the osteoclast and decrease bone resorption. But because bone resorption and bone formation are coupled, you also always see a decrease in bone formation that may limit the efficacy of the anti-resorptive drugs. By contrast, when you treat with a senolytic and kill the senescent cell, you reduce bone resorption, but because you're also having a beneficial effect on the osteoblast, you either have no change or an increase in bone formation. So what I've termed a beneficial uncoupling between bone resorption and bone formation, which may give this approach an advantage over standard anti-resorptive treatment. So in another series of experiments, what we've done is look at the question of frailty and whether senescent cells can induce frailty even at a distance. So fracture results both from low bone mass, but also from increase in frailty. So here, young mice, and remember these are 6-month-old mice, were either given control cells or senescent cells that were made senescent using radiation. And so these were transplanted into the peritoneal cavity, and physical function measurements were made a month later. So this just shows that these cells were made luminescent. They stay in the peritoneal cavity. So whatever effects they're having on frailty or anywhere else is due to systemic effects of the SASP and not because these cells are actually traveling to distant sites. So one of the interesting things was that even a very small number of these senescent cells can have some significant effects. So if you look at grip strength, for example, which is a good marker for frailty, so this is the grip strength in the control mice given PBS or non-senescent cells. And you can see here with 200,000, 500,000, or 1 million senescent cells, as few as 500,000 senescent cells, which is a tiny percentage of the total cells in the mouse, is causing a significant reduction in grip strength, and similarly for some of the other markers. One of the interesting findings from this study was that even though the cells were in the peritoneal cavity, they were actually causing an increase in senescence in the muscle. So here's P16 message in the muscle that went up along with TNF-alpha and IL-6. So these senescent cells can have systemic effects, and that's certainly being looked at with great interest. So we then went to old mice, 20-month-old mice that were treated either with vehicle or with DQ for 4 months. And again, in old mice, again, if you look at grip strength, there's a significant improvement in grip strength, but also in a number of other markers of frailty. But one of the interesting findings was that these mice also lived longer. So post-treatment, the median survival in the control mice was 140 days, whereas if they had clearance of senescent cells with DQ, it was 191 days, and this is a whole life. So in terms of frailty, which would also be expected to impact fracture then, frailty is increased and lifespan is reduced by increased senescent cell burden. Clearing senescent cell burden reduces frailty and increases lifespan. And what I showed you is that there's a feed-forward loop whereby senescent cells induce cellular senescence even in distant tissues. So not only do senescent cells cause harm locally, but they actually also cause harm, at distant sites through the SASP. So this has led to this idea that as we age, there's an increase in the burden of cellular senescence. So you go from, youthful function to where you get induces of senescence. And then as you get towards later in life, you have enough senescent cells that you start getting osteoporosis, impaired vision, all of these diseases that occur with aging, which, if left unchecked, leads to frailty, sarcopenia, and decreased health span, and eventually death. And the hope is that if you intervene here and reduce the burden of senescent cells, that you can maybe move this health span out further so you live longer without these diseases and develop sarcopenia and frailty later. Now, maybe you also extend lifespan, but the hope is that you at least have a more productive life for the time that you do have. Now, based on this, there's a large number of clinical trials that have already started. I just wanted to highlight one. This is one that we've started where we're using in a proof of concept phase 2 study senolytics to see if we can improve bone turnover in older postmenopausal women. And depending on the findings of this study, we may do longer term studies to look at effects on bone density and other parameters. So, that's the positive side. What are the potential concerns of targeting senescent cells? One that's been brought up is cancer. It's been postulated that senescence likely evolved as an anti-cancer mechanism, whereby cells that had accumulated DNA damage and oncogenic insults were redirected towards a senescent growth-arrested phenotype rather than uncontrolled proliferation in cancer. So the cell had to make a choice. Does it become cancer or does it become senescent because it's got all this DNA damage? And so if you prevented this from happening, the concern is, would you have more cancer? But I think there's reasons to believe that this may not be an issue. I think the biggest one is that the senolytic drugs are effective when given intermittently. So these drugs basically periodically reduce the burden of senescent cells rather than preventing them from forming. In our DQ studies in mice, for example, the D and Q have half-lives of hours, and we only give them once a month or twice a month. So these drugs would not prevent these senescent cells from forming, but only periodically reduce the burden of senescent cells. And interestingly, there's also data that if you have cancer and there are senescent cells within the cancer, these cells, because they secrete such a pro-inflammatory SASP, they actually promote cancer growth, which is deleterious. A second concern is in development and wound healing. So it turns out that senescence is a normal phenomenon during development. So developmentally programmed cell senescence has in fact been identified at multiple locations during mammalian embryonic development. And in terms of bone, it's of interest that programmed cell senescence occurs in the primary spongiosa of long bones in mice at the growth plate. And so that in part contributes to the cessation of growth, the senescence of these the precursor cells. There's also evidence that cellular senescence may play a role in wound healing. So these are data from Dr. Campisi's group using mice. It's a different mouse model. It's called the 3MR model, but when these mice are given ganciclovir, you clear senescent cells. So there's many different mice here, but let's compare. This is a skin wound. And so these are wild type mice given PBS, or you can look at the 3MR mice given PBS. So this is the wound size. It goes from 100% to complete healing in about 12 days. Now, in these mice, if you clear the senescent cells by giving ganciclovir, what these investigators showed was that there was a delay in the skin wound healing. The wound did eventually heal, but the time course was delayed. And in mechanistic studies, what they showed that when you have injury, there's accumulation of senescent cells. So these cells become these senescent fibroblasts, and they secrete PDGF alpha, which then recruits fibroblasts to close the wound. So if you get rid of these cells, they found that you don't have enough PDGF alpha and you have an increase in the wound. So obviously this was of concern to us. So we asked the question, what would happen if you clear senescent cells using fracture in a mouse model using D plus Q? And this is just the study. The fracture was induced in the mice. They were then given intermittently D plus Q on a weekly basis, and we measured a number of endpoints. So you can see that D plus Q worked in the fracture site, where we showed in this paper that there is in fact an accumulation of senescent cells, just like in the skin. The D plus Q reduced the P16 message, the PDGF alpha message, and FOXO4. These are all markers of senescence. But in contrast to the skin, where skin healing was impaired, with clearing of senescent cells in fracture, it was actually accelerated. So you can see this is the callus area weeks after fracture. And you can see in the mice given D plus Q, they actually had a more early development of the callus, which eventually started to get resorbed. And if you measure the area under the curve, it was significantly higher in the mice with fracture who were treated with D plus Q. So bone appears to be quite different from skin in terms of the role of senescent cells in healing. This just shows the micro CT. So again, with D plus Q versus vehicle, there's a higher callus bone volume. And if you do mechanical testing, this is torsional testing, D plus Q actually resulted in a stronger fracture, a stronger healed fracture at the end of the treatment period. This is a similar trend for the strength. So what these studies said is that senescent cells do in fact accumulate transiently in the fracture callus as they do in the skin. But in contrast to the skin, clearance of senescent cells in the fracture does not impair, but rather actually improves fracture healing. This obviously has clinical implications for the development of senolytic therapies for osteoporosis, because if we had found that senolytic treatment made it more difficult to heal a fracture, you really couldn't use these drugs in osteoporosis. But fortunately, we found that in fact, it improves fracture healing. But it also has some biological relevance for our concept of senescent cells as uniformly facilitating healing, because these beneficial versus detrimental effects of senescent cells on injury repair may be different across different tissues. You know, in this case, skin being clearly different from bone. So what are the factors that modulate senescence? I'm often asked, you know, what can you do? Should you take senolytic drugs? And I would say I wouldn't take drugs at this point until we learn more. But there are lifestyle measures. So we know that senescent cell burden in mice and in humans goes up with aging. It goes up with a high-fat diet. They go up with physical inactivity, with smoking, with chemotherapy, with radiation, with chronic HIV infection. Now, obviously, we can't do anything about aging, but many of these other things, perhaps, you know, you can't stop necessarily from getting cancer, although some are preventable, but avoiding these things that increase senescent cell burden is a useful approach. And on the positive side, we know that regular exercise and caloric restriction, in fact, decrease senescent cell burden, both in mice and in humans. This is actually a nice meta-analysis of the effects of exercise on senescent cells. This is measured in T cells. And you can see they did a meta-analysis of 3 different studies. And in each study, the P16 mRNA in the T cells was reduced. And overall, you can see almost a 40% reduction in senescent T cells using this assay following exercise training in humans. So exercise certainly is a great way to reduce your senescent cell burden. So I want to close now by acknowledging the people in my lab, my collaborations with the Kirkland lab, Labrasser, many other groups at Mayo. We also received funding from the National Institute on Aging, from NIAMS, and from Mayo Foundation. And I'll end there. Thank you.